Good evening, ladies and gentlemen. It is my proud privilege to welcome you for this webinar on BIAPEN. This is first time in India BIAPEN being launched on the virtual platform, and we have amazing faculty from India. And you will be glad to know that we have 1100 plus delegates registered for this webinar in a very short period of time in 2 days. That shows the interest about the uh, molecule. And these 1100 registrations have come from uh, 500 different institutes across India from 77 cities. Not just in India, but we have got registrations from 10 more countries as well. Uh, can you go to the next slide? A little introduction of BDR Pharmaceuticals. BDR, a research-driven organization established in 2003, and BDR specializes in early identification, development, and introduction of new medicine for life-threatening diseases. BDR is recognized as a niche player in manufacturing of pharmaceutical API for renowned newest formulation. BDR group of companies have grown steadily over last 1.5 decade in both domestic and international arenas. Group is complemented majorly by two companies, BDR Pharmaceutical International and BDR Life Sciences. Each one has stamped its own supremacy in respective fields, that is API and formulation manufacturing respectively. BDR Group focuses on development of 6 specialized therapeutic segments: oncology, critical care, gynecology, neurology, dermatology, and women's health. Next slide, please. BDR has been at the forefront of developing the new molecules and launching it for the first time in India. So many generics they have brought in over last couple of years. The list includes phosphomycin, Anidulafungin, micafungin, cascofungin, daptomycin, doripenem, doliconazole, dagaralex, rucaparib, nintedanib, enzalutamide, clofirabine. It's a long list and impressive. So that shows a fast pace at which backend team is working to develop. In fact, they are forefront. The BDR team has been at the forefront of in the fight against COVID-19 infections in India and globally. With a great speed, they have launched remdesivir, favipiravir, and liposomal amphotericin B in fight against COVID-19. BDI has a strong presence in markets such as North America, South America, Europe, Africa, and Asia. BDR is committed to pass on the benefit of global exposure to the doctors by offering latest life-saving therapies at an affordable price for the Indian patient. BDR is committed to make affordable healthcare a reality. So, next slide, please. With this, we would like to begin our webinar. It is my privilege to welcome and introduce Dr. Shrikant Sahasrabudhe, our moderator and speaker. Dr. Sahasrabudhe is Director and Head Department of Pulmonology and Critical Care Medicine at Medicover Hospitals, Aurangabad. Dr. Sahasrabudhe has over 18 years of experience of managing multi-specialty ICU as in charge and director ICU at Bajaj Hospital. His areas of interest are sepsis, mechanical ventilation, antibiotic stewardship, fluid and electrolytes, rational use of antibiotics, and COVID critical care. Welcome, Dr. Sahasrabudhe. Over to you.
Yeah, so thank you for those kind words. And I'm thankful to the BDR company, especially Mr. Sujay Kale, the general manager who gave me the opportunity for today's session. And I am equally delighted, and it's my honor and privilege to introduce our chairperson for today's CME, Dr. Yatin Mehta sir. Hello Mehta sir. And he doesn't need any formal introduction, and he was the one who said, I don't want a formal long introduction, but I can't stop myself, sir. I think people should know about the achievements you have got, and you're a torchbearer for our society and for entire medical fraternity as far as critical care is concerned. So Mehta sir is the chairman and chairman of the critical care, pain management and anesthesia department at Medanta Medicity, Gurgaon. He has over about 40 years experience in the same field, and his areas of expertise includes cardiac anesthesia, critical care, pain management, and cardiopulmonary resuscitation. Uh, Dr. Mehta was awarded the FIA CTA, that is the honorary award, and also the fellow in the TE by Indian Association of Cardiovascular and Thoracic Anesthesiology in 2019, FICCM by Indian College of Critical Care Medicine. He's also a national coordinator for Dr. Rosenthal's International Nosocomial Infection Control Consortium. And in addition to this, he is on the editorial board of several journals and has about 200 publications and 250 presentations to his credit. He was the vice president of ISCCM Delhi branch and also has been executive committee member for the 2 terms, and of course as a president of ISCCM, which he was uh, uh, in the recent past. He was also additionally he is a president of Indian Association of Cardiovascular Thoracic Anesthesiology. So it's my honor and pleasure, sir, At the same time, I would like to introduce our international speaker, Dr. Abhinay Gupta, who is presently as a consultant intensive care at Homerton University Hospital, London. He was additional medical superintendent and head of the critical care and emergency department at Shada University, Greater Noida. Uh, he has a research as a researcher in as a principal investor at Homerton for genomic trial and Sandman. Uh, at the same time, he was a co-investigator for the famous REMAP-CAP trial, which I think everyone is aware of as of now. And he has several national and international publications along with the authors in couple of the books. So with this, I think I'll start my presentation for today. Now, just to give you a brief and just a thought on this, that as we are aware uh, that there is about about 10 to 15 years of the gap in between the introduction of a new antibiotic as such, especially the carbapenems. And the second generation carbapenem, we had a significant dearth as far as the new agent is concerned. And uh, we as a critical care community are always perplexed by the over increasing multi-drug resistant infection in the ICUs. And if we have a agent which is in the form of a BIAPEN coming up, I think we see some light on the horizon to fight against the multi-drug resistant gram negative microorganisms. And just a thought and kind of one more important information regarding BIAPENM, that it has a better pseudomonal activity against the pseudomonas as compared to meropenem. So with this kind of a short overview, I'll just go on to actually general overview for BIAPENM, where it is a new parenteral carbapenem antibacterial agent with a broad spectrum, as I just now said, of the in vitro antibacterial activity, and it encompasses many of the gram negative organisms predominantly, and the gram positive aerobic and the anaerobic bacteria, which includes the species producing the beta lactamases. And we know that there are so many organisms which produces the enzymes, be it be a beta lactamase or NDM producing or OXA producing organisms, which is the biggest threat to the mankind as of now. So the mean plasma half-life of this agent is about an hour, and it combines with the penicillin binding proteins and inhibits the bacterial cell wall synthesis. Now, as far as the existing therapy is concerned, as we know that we have uh, meropenem, imipenem, doripenem, and ertapenem as one of the carbapenems, the list of carbapenems which we have as of now. So if we compare the innovation versus the existing therapy and the earlier carbapenems like imipenem, which are not that stable to the hydrolysis by the human renal dihydropeptidase, that is DHP1, and therefore it is co-administered with the DHP1 inhibitor or cilastatin. So you need to actually combine imipenem with something which will make it more stable. As compared to that, BIAPENM is a newer parenteral carbapenem, which, unlike imipenem, has one beta methyl group at the C1 position, conferring the stability to the hydrolysis by the human renal DHP1. So it only means that it is more stable than the existing carbapenem, and it can be kind of used in the multi-drug resistant gram negative sepsis. So in vitro investigations, BIAPENM was more stable, as I just now said, as compared to the other penems, and it is administered as a single agent without combination of any other agent for it to make more stable. Now, it has better efficacy where the studies have shown that the efficacy rates for BIAPENM can be as high as 89% as compared to 73% and 86% for imipenem, cilastatin, and meropenem, respectively. As far as the activity against Pseudomonas and Enterobacterus is concerned, and I just now said that BIAPENM, which is fourfold more active against Pseudomonas and is highly superior to imipenem cilastatin against the Enterobacteriaceae. So we see the infections in ICU caused by the Enterobacteriaceae group, mainly the Klebsiella and the E. coli, which are becoming more and more resistant, along with the other gram-negative organisms like Pseudomonas and the omnipresent Acinetobacter. So these are probably the four more common organisms which we come across. As far as safety profile is concerned, it is almost non-toxic to kidneys and the central nervous system. So the major hurdle to use such products in the patients who have multi-organ failure, which will have the advantage over the other penoms. As far as the resistance to hydrolysis is concerned, and it has been already said that it is more stable, and the more important point of less frequent dosing, and the less dosing by far, you can have 300 milligrams twice a day because it is widely distributed and penetrates into the various tissues and the body fluids, and also has a long post antibiotic effect. So these are few of the advantages of the BIAPENM as compared to the other available penoms. It is well tolerated, and in comparative study with less adverse events with the 300 milligrams twice a day dose, with highly efficient less dose, which is compared to other penoms. Like if you see meropenem, you have to give it about 3 grams in a day as compared to about only 600 milligrams of BIAPENM in a day. So these are the common adverse events which were seen, like skin eruptions, rashes, nausea, and diarrhea, with increased ALT and AST levels, with mild eosinophilia and decreased creatinine clearance, which were commonly reported side effects. As far as indications of BIAPENM is concerned, there are standard indications like sepsis or septic shock, the complicated urinary tract infections, even the intra-abdominal infections form the important indication, along with the complicated lower respiratory tract infections. So these are probably the four common areas where we face the challenge of using or treating the patients. As I already said, the recommended doses of BIAPENM is about 300 milligrams every 12th hourly in the intravenous form for the period of 3, 7 to 14 days, depending on the severity of the infection. And this is what I said, meropenem, we need to give it at about 3 grams per day, and imipenem cilastatin of about 500 to 500 ratio of imipenem cilastatin every 6th hour. So at least 2 grams has to be given. And in severe cases, we may actually go up to 1000 milligrams every 8 hourly. So it comes to about 3 grams of meropenem and imipenem each. Just having the thoughts which have been put forward, I would again like to repeat the key points for BIAPENM, which is less in the dosage as compared to the other carbapenems, the drug resistant profile for BIAPENM, which is reasonably less as of now, and the stability of this particular drug against the hydrolysis by the DHP makes it different from the other carbapenems. So these are some of the points which I thought I would deliberate today and take this help of this platform to sensitize our critical care colleagues across the globe as to how useful can this product be. So I think with this talk, I would like to request our international speaker, Mr. Abhinav Gupta. Hello, sir.
Hello, hi. Thanks,
Yeah,
Shrikant. Thank you
yeah,
very much for the kind introduction,
yeah.
and thanks for remembering and inviting me. My regards to my teacher, Dr. Yatin
Yes,
Mehta, with whom I've been had been a student and later on worked with him for such a long time.
Yes.
Regards.
So,
Thank you, sir.
so very nice to have you. And may
Thank
I
you.
please request you to deliberate your talk?
Sure, thank you. So basically, I'll be sharing my presentation actually, and then I hope I'm able to do it actually. Otherwise, I'll have to request the organizers to share the screen to get the presentation up, please. Um, I think I just had to change my computer. Something went wrong with the computer. I'm sorry about that. And um, I think I'll request the organizer to share the screen, please. Share the presentation, please. Thank you. So that's useful. So I've been asked to talk on BIAPEN. I have no financial disclosures at the moment, and BIAPEN is, to be honest, we have not used in NHS, but it appeared to be a nice and promising molecule. BIAPEN. Next one, please. And we've already learned more about BIAPEN by Shrikant, who's who's talked about BIAPEN in great detail, and it appears to be a new kid on the block, addition to the whole lot of carbapenems. And thankfully we are still working on introducing new antibiotics, basically because as we all know, we are struggling with infections and antibiotics are in short supply. New antibiotics are not in much supply at the moment, so hopefully it comes up with some promises, and it has greater antibacterial activities with a more wider range of against gram-positive and gram-negative bacteria. This is what we've been informed, and especially against Pseudomonas aeruginosa and Bacteroides species, which is probably more than imipenem, but I would not like to commit to that because there are still a lot of studies going on. I was reading more about it in the morning and about head-on trials, and let's wait for more trials to come. But at the moment, what I could gather is it has a wide range of antibacterial activities and may be worth a try. Next slide, please. So the susceptible strains are both gram-positive and gram-negative organisms. Uh, the next slide, please. Thank you. So they've mentioned a head-on trial comparison with imipenem and meropenem, and it is more potent. The dose is twice daily as compared to a thrice daily dose of meropenem and a 6-hourly dose of imipenem celestin combination. And it has bactericidal activity against biofilm formation and resistant PA as compared to imipenem and meropenem, and what they've documented is an excellent in vitro activity against Acinetobacter. So now the point is that any additional benefit will get, it's just for time to see. But the only thing is, presumably they should in an effectively there should be less resistant against biopenem as compared to imipenem and meropenem, considering that it is a neo drug. Next slide, please. So in short, it is the key point. It has got a powerful action against Pseudomonas. It is widely distributed, penetrates well into various tissues, especially lung tissues, because there are studies regarding its penetration in lung tissues and its effectiveness against pathogens in lungs, which is quite encouraging, and in body fluids as well. And the advantage is obviously twice a day dosage with a broad spectrum coverage. I I would still suggest that please go for the microbiological spectrum before you start using it empirically. But again, worth a thought, and the safety profile is still to be ascertained in further studies to come. But it is a promising molecule that might, and we have to see about how much progress it makes. Thank you. That's the end of my presentation. Thanks very much, and wish everyone a wonderful evening and a great success. Thank you.
Yeah. So I think I would again thank Dr. Gupta for such a presentation. And with this, if I may request Uh, Dr. Yatin Mehta, sir, if we can take few of the questions.
Sure.
Am I audible?
Yes, yes, absolutely.
Yeah. So I think if we can take few questions, sir, with your permission.
Yeah, sure.
Yeah. So BIAPENUM is a new carbapenem. So what can you say? What can you have a comment? Anything more than uh, about the this molecule?
See, Shrikant, I see a question in the Q&A which says how bipenem is superior
Superior.
than meropenem. Well, that is again, as Abhinav rightly said, I think it is too early to say that because most of the papers are Asian papers, Japanese papers, because that is where it was, and I've read a few Thai papers. So it is approved in Japan, China, Thailand, and now in India. So it is a little difficult to say that. But what major advantages I find is that this patients who are particularly uh, susceptible to seizures. I think obviously imipenem has the highest, possibility of causing seizures. Seizure threshold is lowest for imipenem, and then it is for mirapenem. So if a patient has seizures or is possibly likely to get seizures for whatever pathology he has, then probably bipenem is superior because bipenem has not been reported to produce seizures. That is one. Secondly, obviously, it is twice a day dosages, so it is more convenient to give. Imipenem mostly is 6 times, 4 times a day, 6 hourly, and meropenem is 3 hourly. So that is another thing. Third thing is that it's a relatively new drug. It's a new drug in India, at least. So probably the bacteria have not seen the drug. So they probably may not have resistance. So possibly any new antibiotic will, at least for the initial period, will have better resistance than the lower resistance than the existent ones which are being used. Meropenem and imipenem, as we know, are extensively used. Also, one advantage would also be about the DHP, which you mentioned, that imipenem is combined with cilastatin. But I think that is a relatively uh, lesser of an advantage because meropenem does not really require that. I mean, DHP1 inhibitor. So that is not a problem with meropenem. It has been used quite a lot of publications are there for all cavitary. It has been used for intrathoracic, that means pneumonias. It has been used for complicated intra-abdominal sepsis and complicated urosepsis. So these are the indications for which one can use it. So there are some certain advantages. Um, Theoretically, it has been shown some pseudomonas and all has been shown an Acinetobacter to be less resistance to this than meropenem. But most of the difference is either they're equal or they are marginally different. So it is maybe
Or,
too early to really.
or not superior to the other drug, maybe some kind of.
Cost may be an issue because in sicker patient we use 2 grams meropenem 3 times a day. That compared to this 2 times a day, 300 milligram normally, which is the standard dose, may be an advantage.
Yeah, thank you, sir. And just a bit, it has been said about the antibacterial activity a bit, but can you please comment on its antibacterial activity, kind of a broad spectrum which we talk of? So,
See, it is effective against most of the gram-negative bacteria. So it is effective against Klebsiella, it is effective against E. coli, other Enterobacteriaceae, Enterobacter, Pseudomonas, and Acinetobacter. As I said, Pseudomonas and Acinetobacter, the sensitivity in a few studies is better than existent carbapenems which are in use. It is also effective against gram-positive bacteria, that means except for MRSA. So it is effective, mostly MSSA. It is effective against Staphylococcus, MSSA, Streptococcus, Haemophilus influenzae. Uh, so it is quite effective against that. It's also effective against bacteroids. I mean, anaerobes.
Right.
So it will be effective against bacteroids, streptococcus, and so on. So spectrum is quite wide. Does not work against MRSA. So that one should be clear about.
That's a very that's an important point which especially our surgeon friends tend to forget. You know, they are very fond of adding metronidazole to a carbapenem, whereas you correctly said that if they cover anaerobes, probably we need not add any anti-anaerobic cover for the patient. So I think the indications have been said, and I don't think we need any further clarification on that. Probably the urinary tract infection, as you said, intrathoracic infections, intra-abdominal infections, and sepsis per se, as a or a occult sepsis where we have a patient with a septic shock. Probably this can be used. And anything about the dosage, sir? Because there's a comparatively, as you say, that meropenem we can use up to even about 6 grams in a day. Vis-a-vis, this is a less dose. So any comment on that?
See, normally 300 milligram twice a day is what is the dose recommended for this. Up to 1.2 grams a day has been used. Meropenem, as I said, in septic patients, very sick patients now, most of us are using 2 grams 3 times a day. But that is that. Also about imipenem silastane. Most of us would be using 1 gram 6-hourly imipenem, for sicker patients. Whenever I de-escalated the dose to 500, it never works. I had to always jack it up. Although now more and more use meropenem or doripenem rather than imipenem. Anyways.
Right. So there was one more question in the question box
I mean,
about.
no, you're please, you can interrupt any second or please say whatever you want to add. It's nice to see you, Abhinav, after such a long time.
Thank you, sir. Thank you very much. Thank you.
Right. He has extensively worked, and we had his interactions very very actively during the COVID times, and it was a very tough time
See,
to
he is
develop this.
academically one of the best students I've had.
Thank
And
you,
I
sir. Thanks.
really miss him.
It has been an honor to work with you, sir. Thank you. Thank
Okay.
you, sir.
So Dr. Abhinav, as sir said, if you want to add anything onto this.
So basically, just as Dr. Mehta said, Shrikant, is that it's a new carbapenem, and it's joining the other family members of carbapenem, and essentially the spectrum is maybe slightly better, but again the studies need to show. The safety profile is, because it's a new drug, we still have to show we have to still ascertain whether it is very safe to use. I think papers would start coming on very soon about safety profile as well. And to use empirically, I'm not very sure whether you should be jumping on that, but again, go by the microbiological spectrum. We, and as you know, in the UK, it is very tight in terms, because microbiology is a safeguardian of antibiotics, actually. So we have to, any new thing we have to use along with microbiology. It's not that things are quite open here to start any new drug. Yes, it is, they are quite receptive, but it has to go through extensive paperwork, and we have to establish that it is safe to be used. That's the only thing. And so many drugs have come recently, actually. So, and we can definitely propose, if it is being used extensively, yes, this is another thing that we can be used. And as far as resistance is concerned, yes, may have a slight edge over the other carbapenems, basically because it's a new drug, and relatively maybe theoretically should have less resistance. But and again, it's a new drug introduced in India. Basically, extensive studies from Japan and Southeast Asia. So maybe some new profiles and new papers that come from India may establish its more safety, well, its safety profile and its extensive usage. Yeah, thank you.
Yeah. So, sir, anything, Mehta, sir, anything on
Yeah,
the
I
safety?
would like to add to the manufacturers that they'll have to provide the discs. At the moment, I think CLSI also, there are no standard MIC guidelines for this drug. Obviously, it takes a long time, and there's a lot of political reasons not to do it for any drug which is developed in Asia. But I think the manufacturers will have to give us the discs, because without the sensitivity, it is difficult to justify use of an antibiotic.
That's correct. Just extending the same profile of the drug as far as the side effects and the safety profile is concerned. There was one question which has come in the question bank about the dosing in the CKD patients. So uh, I suppose it applies to the same principle applies here, that the studies which we have as of now are like limited as far as the use of BIAPENM in different spectrum is concerned, and it will probably go along with the same principle to even the CKD patients as to how we kind of adjust the dose, as we have already mentioned that the maximum dose of 1.2 grams per kg which can be given. So from I think from my side it would be that we need to actually look at more PKPD studies for this particular drug to comment on the dosage in CKD. Mehta sir, if you can have any
See,
comment.
normally they recommend that patient on a CKD patient with probably a GFR less than 30, GFR or whatever, normally 300 milligram once a day is what needs to be given. But yes, again, there are not enough PKPD studies for in these sort of group of patients.
Yes, correct. Another question which I have here is, does it have a cerebral penetration like meropenem?
Theoretically, both meropenem and imipenem, they have cerebral penetration, and I think the profile would be similar basically. So unless again we have to go by multiple studies that come into being, but theoretically, because imipenem and meropenem and carbapenems, they can penetrate blood-brain barrier. So they should, ideally it should have a cerebral penetration.
Yeah,
But
so
again, we need some more data on that
But
basically.
although I have not seen any publication on meningitis, used to this is
No,
meningitis.
no, I totally appreciate that, sir.
right. So another question is, what are the chances of the cross resistance to other carbapenems?
Well, I would expect it to happen. I mean, most of the, if you look at it, most of the uh, bacteria which are resistant to imipenem would be resistant to meropenem. Maybe doripenem maybe have a little different this thing because whatever reason. But I mean, at the moment, as I said, it's a new drug, so it may not be as much, but the cross resistance would likely to be there.
Correct, absolutely. And the thing is that if we, as you correctly said, sir, and that's a practical point which we face, at least in our part of the world, is about having the disk and having the microbiologist confidence on whether this drug is really sensitive for one particular organism. I think that is an important point. And another thing is whether we can, whether you would be using this drug as an empiric drug or an empiric antibiotic for any patient who comes to us with a septic shock. So just given the whatever the information we have, uh, would we use it upfront as an empiric choice as compared to other penums, or we still probably believe that meropenem would be probably better? Somehow, I think there is another thought process which can go that a drug with a better dosage or with a little high dosage would actually act a little better than the drug with a smaller dosage. I don't know. It's a kind of a very silly question, but then I am not very sure whether we can use it upfront as an empiric choice. So, Mehta sir, your
See,
comment.
that may not be absolutely correct. I mean, the doses are based on PKPD studies, not the more the better. I mean, that may not be correct. Uh, but I think whether we will use it for septic shock, I don't know because I have not seen any studies on septic shock. I've seen papers on sepsis.
Right.
So I think initially it has just been introduced in India. We should start using for patients with sepsis rather than patients with septic shock because septic shock, the, not enough published evidence, and also these patients have a higher mortality. So I would not advise to begin with because again you can get into difficulty if you try a new drug. Uh, which has not been used in septic shock. So sepsis is one we should start with. Then maybe we'll mature later on. What do you say, Abhinav?
So yes, I agree with you, sir. But at the present, our practice is basically the microbiologists guide us from the basis of on the basis of our microbiological profile, and at the moment, our microbiological profile is piperacillin tazobactam is a drug effective enough for all kinds of septic shock at the moment. So we haven't reached a stage that we need to start carbapenem as a first line agent, actually. So as an empiric therapy, we start with piperacillin tazobactam and then reach to meropenem if we have to, actually. And uh, to start with, if I were to make a choice and start a new drug, all of a sudden I would slightly be hesitant. But maybe in years to come, if, with more practice and with more publications coming in, it could be one of the potential drugs to start with as well. Yes, it has maybe it might have a potential.
So as you say, Dr. Gupta, you have a piperacillin tazobactam as a drug which is sensitive to your organisms causing septic shock.
Yes, yes. At the moment, we are very comfortable with piperacillin tazobactam. Actually, you'll be surprised. Actually, you'll be surprised. Our first choice antibiotic is benzyl penicillin. In fact, it's been years now that we've started using it back again. In fact, I've been working in this hospital for last 2 years actually, and we started after 2 years. We are starting again with benzyl penicillin,
Okay,
and then moving on to piperacillin tazobactam if you have to.
correct. So for you, probably using the carbapenem would be like a talk of the town if you really use.
No. That's not the thing. In fact, that's not the thing because, as you know, we still have acinetobacters. We still have all the multi-drug resistant Enterobacteriaceae. But the only thing is, as soon as this patient is diagnosed with something, he's quickly isolated, and so that there's no with all contact precautions. So and I think it is just like any other ICU. It is being used. Carbapenems are being used, but that's our resort, not taken very soon, but at a later stage.
And if I may ask, what are the common organisms who which are actually grown in your ICU?
So that's very interesting, basically, because I personally did an audit on because I was concerned with high infection in the ICU. So I took the audit upon myself, and I did it basically. And so the most common organism that we are growing of is Pseudomonas, followed by Klebsiella, and then going to gram-positive Staphylococcus aureus. So Pseudomonas is definitely the most common organism, and it is at the moment sensitive to tazocin. We call it piperacillin tazobactam actually, and we do have resistant E. coli as well. We do have ESBLs as well, and meropenem is widely used. We are not using imipenem at the moment. We're just sticking on to meropenem if we have to use it. But again, it is different in different hospitals. Some hospitals prefer imipenem. So our hospital prefers meropenem.
No, see, Abhinav, for piperacillin tazobactam for as an anti-pseudomonal is okay. I mean, because it is likely to be
Sure.
sensitive in your. But is the Klebsiella sensitive to piperacillin tazobactam?
Sir, it is most of the gram negatives are sensitive to piperacillin tazobactam at the moment. So we are not growing a lot of ESBLs. But yes, as soon as we go to an ESBL, be it E. coli or Klebsiella, we switch over to meropenem. Yes.
No, but sir, but then theoretically, piperacillin tazobactam should work against ESBL.
Theoretically, yes. Again, but if it is resistant, yes, we move to meropenem.
And how is
Or
your
if the patient is deteriorating clinically, if the clinical deterioration is there despite piperacillin tazobactam, we move to meropenem.
and how is the incidence?
And gentamicin. We use a lot of aminoglycosides. We use a lot of aminoglycosides as a first line therapy in case of resistant gram negative,
Along with pip-tazo also
along with piperacillin tazobactam as well.
as a synergistic.
So our step ladder pattern is we start with comoxiclav or benzyl penicillin, and if the patient is a community acquired, and if the patient is hospital acquired, we start with piperacillin tazobactam as an empiric therapy. And if the patient is clinically deteriorating, we add an aminoglycoside because if it is out of hours, and if the septic screen is still awaited, if the patient is still not improving, then after discussion we need to.
I think Dr. Gupta's connection is little unstable. So if I may ask,
I'm back. I'm back. Sorry.
Yeah, you're back.
Sorry. And then because this last 2 years is entirely different, we are seeing a lot of fungal infections. And if despite antibacterial therapy, we are still struggling with infection, with sepsis and septic shock, we empirically we have started adding anidulafungin or amphotericin B, preferably amphotericin B or voriconazole, both of them.
And how is the incidence of MRSA at your place?
MRSA is very low. Even a single MRSA is a big thing in the trust, and it is counted at a national level. So both C. diff and MRSA incidence is very low at the moment. Fortunately, yeah, touch wood.
So if I can ask Mehta sir, is it a standard practice in your ICU to administer antibiotics, or especially carbapenems or broader spectrum antibiotic, in an infusion or a slow infusion form or as a bolus?
No, nobody, I mean, everybody seems to be using it as an infusion now. That is the standard. It makes PKPD sense.
Right.
So
So,
about 3 hours infusion.
correct. And how is your use about as far as doripenem is concerned, as compared to meropenem and?
Not as much as meropenem, but yes, we do use regularly doripenem also.
Okay. I think as of now, I don't see any other new questions. So if I may request Mehta sir to have, concluding remark.
Yeah. So we have a new kid on the block. I can't call it kid because it is now 18, 20 years old. The drug was in 2002, first time used in Japan.
Right.
It always happens. With any drug which is developed in East, it takes a long time to be recognized by the West. And then, as you well know, even teicoplanin is still not recognized approved by FDA. There are thousands of papers on teicoplanin. Anyway, so I think one should not be, we being Indians don't necessarily have to be skeptical. Also, Japan is not a developing or a third world country. Let me tell the audience. Japan, the healthcare standards are very high. The public health standards are also very high. The microbiological support is excellent. I mean, they're as westernized as any. They don't even consider themselves to be Asians, and from the political point of view. But I have interacted with many Japanese senior physicians, and the standards are very high. And so I think if it is a proven track record in Japan, but we need to just be introduced. So we begin with urosepsis. It has certain advantages, certainly. Um, Easy to administer because twice a day. It may be cheaper. I don't know exactly what pricing they have done in India, but it certainly will be cheaper than high dose meropenem. It is supposed to be more effective against Pseudomonas and Acinetobacter, so that may be an advantage. Particularly, we don't have such a major problem, but public hospitals in India have more incidence of Acinetobacter and Pseudomonas. And it has been shown, I mean, repeatedly that Acinetobacter not only is mostly MDR, it's also a issue of transmissibility in your own hospital environment because of the nursing care may not be good. So that's why it's more so public hospital. And Acinetobacter patients are likely to have MDR organisms and likely to die also more. I mean, so these are the problems. So this may be an advantage. But again, these are all still unproven question marks, which we have to use the drug and then see how it goes. New drug obviously would likely to be the bacteria may not develop resistance that fast. It may happen over a period of time. So maybe you'll find better sensitivity initially. So these are the things which we have to look at. We can have an open mind, and I'm likely to start using it soon, at least in patients who are not dying or don't have huge Apache or SOFA scores to begin with, and then we'll see. But it is not a wonder drug, but there are possibilities with this newer carbapenem.
Okay, thank you very much, Mehta sir and Dr. Gupta. And I would hand over it to Mr. Rajesh.
Thank you very much, Dr. Sasrabudhe, Dr. Yatin Mehta sir. and Dr. Abhinav Gupta for your deliberation. I would like to request AV team to play the launch video. Thank you, team. With this, I would also like to thank Mr. Shailesh Thare, Mr. Sujay Kale for bringing this quickly with us, and Mr. Sameer Shah. Sir. Thank you very much, all delegates who have attended it. Thank you.
Thank you. Thanks very much. Thank you, sir.
Thank you. Bye.
Thank you.